Video will continue section 8.9 on osmosis and dialysis, but we will focus on the human body in this part of the section. So, two solutions with the same osmotic pressure are said to be isotonic, which means they have the same number of solute particles dissolved in them. So in isotonic, there is the prefix iso, which means the same, and tonic refers to tonicity. And tonicity, you can see that as it's almost a synonymous to a concentration. So same concentration of solute particles in the two solutions. But remember that osmosis is a colligative property that depends on the number of solute particles dissolved, and it does not matter the identity of the solute particles, just that here when it's isotonic that the number of solute particles is the same between two solutions. So here we have an example of a red blood cell placed into a beaker into an isotonic solution. So that means the solution inside the red blood cell and outside the red blood cell are isotonic, which means they have the same number of particles dissolved in them. And so solutions isotonic to the body are, and they are always the same for everybody. So there are different ways of giving their concentrations, right? Because there are so many concentration units. And so one of the way to say an isotonic solution to the body, it's by saying 0.92% mass per volume percent of NaCl. So a solution of that concentration would be isotonic to the body fluids. Another unit would be to say 0.15 molar NaCl solution would also be isotonic to the body fluids. If we're using glucose, the concentration would be a 5% mass per volume percent of glucose solution would be isotonic to the body. So remember how osmosis is a colligative property, which means it doesn't depend on the identity of the solute. So that's why you can use glucose or you can use NaCl to describe an isotonic solution to the body. But you have to adapt the concentration. That's why when it's the same concentration unit of percent by mass per volume, it's 5% if it's a glucose solution, and it's 0.92% if it's a NaCl solution. And so when a red blood cell is placed into an isotonic solution, it will be happy and healthy and will keep its biconcave disc shape, right? The red blood cells have a particular shape and it will keep that shape and be healthy. Because the two arrows here are symbolizing the fact that water will move out and will move into the cell because, you know, there is always a dynamic equilibrium where water keeps, you know, coming out of the cell and then coming in, but it comes out and comes in at the same rate. So that's why the volume of the red blood cell does not change. Now, if you put a red blood cell into an hypotonic solution, it means this hypotonic solution has a lower osmotic pressure than body fluids. Lower osmotic pressure means the concentration of the particles outside the cell, you know, the solution the cell is placed in, is lower than the concentration of the particles inside the cell. So we have a difference in concentration and it's smaller outside than inside. And so because of that, and because the solutes inside the cell are not allowed to come out, only water can move in and out. And so the water will then diffuse into the cell because it goes from low concentration of solute to high concentration of solute, and here it's less concentrated outside the cell, and so it will move from outside to inside because it's more concentrated inside the cell. And so water diffuses into the cell, and so the cell will swell. Its volume will increase, and if the difference between the concentration is very large, it can eventually burst. And so for red blood cells, this swelling and rupture is called hemolysis. Hemo refers to red blood cells, and lysis means breaking, rupturing. So it really tells that it breaks apart the cell because it bursts. So an hypotonic solution, to remember what it means, hypo here means less than, so the tonicity is less than the tonicity of the body fluids. Now, an hypertonic solution has a higher osmotic pressure than the body fluids. So hyper means it's more tonic than the body fluids, which means it's more concentrated. So the concentration of the particles outside the cell is higher than the concentration of the particles inside the cell. And so again, because the particles cannot move in and out, and only water can, it's the water that will move to try to equilibrate, balance the concentration. And because water will go from a low solute concentration to high solute concentration, it will go from inside the cell to outside, where it's more concentrated, right? Because it's more concentrated outside, so water will move out of the cell, and that means the volume of the cell will decrease. The cell will shrink. It will shrivel. And this process is called crenation because it looks like the cell becomes crenated. So that's when you have a red blood cell into an hypertonic solution. So those terms, isotonic, hypotonic, and hypertonic, are always referring to a cell in comparison to are referring to a solution in comparison to a reference solution. It's always compared to a reference solution. And here the reference was the body fluids, which are the intracellular fluids inside the red blood cell. So we want you to think about this and tell us what happens to a red blood cell when it is placed in each of the following solutions. So let's say your red blood cell is placed into a 3% mass per volume glucose solution. So let me remind you here what are the concentrations that are isotonic to the body fluids. And so solutions isotonic to body fluids are 5% mass per volume glucose solutions, or 0.92% mass per volume NaCl solution, or a 0.15 molar NaCl solution. So when you place this red blood cell into a 3% mass per volume glucose solution, you want to compare the concentration of the solution you're placing your red blood cell in with a comparable concentration inside the body. So you want the units of concentration to be the same so that you can compare them. So then you will want to take the intracellular fluid of your red blood cell as 5% per mass per volume glucose solution, so that you can compare to the 3% mass per volume glucose solution, right? So we will then take the fact that our red blood cell, inside it, it's a 5% mass per volume glucose solution. So if you place that red blood cell into a 3% glucose solution, you see that the percentage, the concentration, is smaller on the outside of the cell than inside. So if it's smaller, the water will want to move into the cell. So first, this would be called an hypotonic solution, right? Because it's less concentrated than the intracellular fluids. And so water will move from low concentration of solute to high concentration. So that means it will go from the 3% glucose solution into the red blood cell to try to balance the concentrations. And so water will move into the cells and swell them. Right? So a 3% glucose solution is less concentrated than a 5% by mass per volume glucose solution, and so it's considered hypotonic. And so water will flow into the red blood cells and swell them. Let's think about what will happen when you place a red blood cell into a 0.15 molar KCl solution. So again, you want to compare this concentration to a concentration of the same unit. So you want to take out of those three isotonic solutions to the body, you want to choose the one that has the same concentration than your 0.15 molar KCl solution. And that means you will want to use the 0.15 molar NaCl solution. So now you have to tell yourself your red blood cell has a 0.15 molar NaCl solution inside, right? The intracellular fluid has this concentration. Now compare it to the 0.15 molar KCl solution. And remember that when you have ionic compound, you need to take into account that when they are in solutions, they are dissociated into their ions. And here in NaCl, we have Na plus and Cl minus, right? 2 ions per formula. Now, if we look at KCl, we will have K plus and Cl minus, one of each. So 2 particles per formula. And because the molarities are the same, 0.15 molar and 0.15 molar, in each case, we will multiply by 2 particles. So that means these 2 solutions are the same tonicity, if you will. So the 0.15 molar KCl solution is isotonic to the intracellular fluids of the red blood cell. And so if it's isotonic, there will be no movement of water more in one direction than the other. Right? So water will not flow more in one, more in one direction than the other. And that means the red blood cell will be healthy in that 0.15 molar KCl solution. So now let's take the case where you place your red blood cell into a 0.15 molar Na2CO3 solution. So again, you want to compare this to the same type of concentration unit and its molarity again. So we will compare it to 0.15 molar NaCl solution. So inside the red blood cell, we have 0.15 molar NaCl solution. And outside, we have 0.15 molar Na2CO3. So we said earlier that NaCl dissolves into 2 particles. So times 0.15 molar, it's going to be equivalent to 0.30 molar, right? And here, 0.15 molar Na2CO3, you need to think about how Na2CO3 will dissociate into solution. And here we have 2 Na plus. So Na is Na plus, and there is the subscript 2 here that says you have 2 Na plus for 1 CO3 ion. The CO3 is the carbonate ion, and its formula is -2, so CO3 -2. So it's a polyatomic ion with 2 negative charges. So we have one one carbonate ion and 2 sodium ions, and that's 3 particles per formula. So that means multiplied by 0.15 molar times 3, it's 0.45 molar, molarity equivalent, right? So it's more concentrated than the 0.15 molar NaCl because you have 3 particles per formula versus 2 particles per formula. Starting with the same 0.15 molar value, that means it's going to be more concentrated. There will be more particles in the 0.15 molar Na2CO3. So it's hypertonic to the intracellular fluids of your red blood cell. So the water will want to move from low solute concentration to high solute concentration. So water will go from inside the cell and move out to try to dilute the more concentrated outside solution. So water will move out of the red blood cell to the outside. And so the cells will have their volume be reduced and it will shrink them. So that's what happens when you place them in a hypertonic solution. So one last word now on dialysis. So in the human body, blood is filtered through the kidneys by the process of dialysis. And so the kidneys is made of millions of nephrons that filter out small molecules and ions from the blood. And then useful materials like water and glucose or ions are reabsorbed later on. But waste like urea or other waste product will be eliminated. And so also in dialysis, so water, small molecules and ions can pass through the semipermeable membrane, the dialyzing membrane, which are the nephrons in the kidney. And large biological molecules like proteins and starch molecules cannot. So that's why it's a very efficient system. It keeps all the big, nutritive molecules inside the blood. And because it takes out water and ions which are needed, these are reabsorbed later on, in the process. But this allows waste products like urea to be removed from the bloodstream, and then they are eliminated in the urine. So when a person's kidney are incapable of removing waste products from the blood, hemodialysis is used. And so the persons who have to go through hemodialysis, they go to the hospital 2 or 3 times per week to have their blood filtered. And so there are machines for that. And so the blood is taken out of the person's body and it goes through a tube. And then there is a portion of the tube that is dialysis tubing, which is a cellophane membrane, and then it's suspended into an isotonic solution that contains NaCl, KCl, NaHCO3, and glucose. And so the reason why the blood, once it is in the dialysis tubing, it goes through these isotonic fluids. It's in order to keep, NaCl, KCl, NaHCO3, and glucose in the blood, because if the concentrations of these particles are the same inside the blood and outside, then they will not move in or out. But what will move out and what is wanted to be moved out are waste, like urea. So these molecules, because they are not contained in the fluid, they will move out of the blood by diffusion, right? The dialysis tubing allows them to pass through, right? The pores are small enough or big enough for the urea and all the waste product to move out of the blood. And so this dialysis fluid is refreshed, you know, to remove the fluid that has been contaminated with waste, so that and so that more bloods can be filtered. And so once the blood is cleaned this way, then it's returned into the body, right? And so that ends section 8.9 on dialysis.